You're much welcome to this session at Medical Sciences. And today's session we want to discuss about mechanism of action of antibiotics. And majorly we want to look at antibacterials, those ones that target against bacteria. So we want to discuss how their mode and then the mechanism. What's the difference between the mode and the mechanism? Mode is the final outcome, whereas the mechanism is the how it happens. And as we begin, always remember to subscribe, hit the like button, and share to others. And as we begin, there are major 4 mechanisms of action of different antibacterial agents. And these include, whereby in number 1, we shall look at those ones that inhibit cell wall synthesis, inhibitors of cell wall synthesis. Then number 2, we shall look at the antibiotics or the antibacterial agents that inhibit the functioning. These ones inhibit the functioning of cell membrane. Then we shall see another mechanism or another mode, number 3, whereby we shall look at the antimicrobial agents or antibacterial agents that inhibit nucleic acid synthesis, nucleic acid, that is DNA or RNA synthesis. Then in number 4, we shall see those that inhibitors of protein synthesis inhibitors. So number 4, we shall discuss about protein synthesis inhibitors. So these are the modes or the mechanisms of action we want to discuss in this session. And as we begin with number 1, inhibitors of cell wall synthesis. We know that the bacterial cell is made up of peptidoglycan layer. So we're going to see those enzymes. In case we have a cell wall, it is surrounded by the cell wall, and this cell wall is made up of these ones, we are calling them peptidoglycan layers. So these are the peptidoglycan layers. And these peptidoglycan layers are always interlinked, or they are cross-linked with each other using what we call penicillin binding proteins. You can call them penicillin binding proteins, or you can call them so this process is known as transpeptidation, whereby transpeptidase is the enzyme that catalyzes the cross-linking of different peptidoglycan layers. So we're going to see the ones that inhibit cell wall, they target penicillin binding protein, or this penicillin binding protein. So we are saying that these inhibitors, they bind and inhibit, they bind to penicillin binding proteins, and you can call them transpeptidase. This enzyme, you can call it transpeptidase. And after binding to it, they inhibit. Hence, they inhibit or they inhibit cross-linkage of peptidoglycan layers. And whenever there is no cross-linkage of these peptidoglycan layers, so what these penicillin binding protein, they normally come together and cross-link these 2 different layers of peptidoglycan. So in case we have taken a drug like penicillin, which binds to penicillin binding protein, what we are calling transpeptidase enzyme, it does not allow the cross-linkage of these 2. And once there is no cross-linkage, the cell wall remains with the pores. So this one creates pores in the cell. And when the pores remain in the cell, there is leakage of intracellular content. So the intracellular content in the cell starts leaking due to the presence of pores in the cell wall due to failure of cross-linkage, leading to cell lysis. So this is how these drugs inhibit cell wall synthesis. And they include drugs that inhibit cell wall synthesis. I can put them here. They include beta-lactams, whereby under beta-lactams we shall see penicillins, we shall see cephalosporins, which have different generation, first generation, second generation. We can see monobactams and carbapenems, like mepenem, meropenem, all of these ones which are broad spectrum antibiotics inhibiting cell wall synthesis. So number one is beta-lactams. These ones, all of them have the beta-lactam ring, and other non-beta-lactam ring include vancomycin. We have fosfomycin. Though vancomycin and fosfomycin, for them, they don't bind directly to the protein, penicillin binding proteins. For them, they inhibit the transglycosylation, what we call transglycosylation. But in the end, they are inhibiting cell wall synthesis. So this is class number one, which are cell wall inhibitors, which we have given example of beta-lactams, then the vancomycin, fosfomycin as also examples, whereby under beta-lactams we see penicillins, cephalosporins, monobactams, and carbapenems. And under penicillins, we have different examples of drugs like natural penicillins like Pen-G and Pen-V, then the semi-synthetic like ampicillin and amoxicillin, then others like ticagrelin, piperacillin, and many others, doing the work of cell wall synthesis inhibition. The mechanism number two we can discuss in this session is inhibitors of the functioning of the cell membrane. And we are seeing apart from having the cell wall, they also have this cell membrane in their structure. And this cell membrane is the one that enables transportation of materials inside, in, and out of the cell. So whenever drugs inhibit the functioning of the cell membrane, drugs here we can see daptomycin, we can see drugs like polymyxin B, polymyxin drugs, and some antifungals like amphotericin. Amphotericin, though it is an antifungal, amphotericin B, it acts as an inhibitor of cell membrane. So these ones, for them, they act by inhibiting. So once they bind to the cell membrane, they makes it or they render it non-functional. And examples we are giving daptomycin, polymyxin, that is polymyxin A, and polymyxin B, plus the amphotericin B, which is an antifungal. It is an antimicrobial agent, which also inhibits cell membrane. Then number three, we can look at nucleic acid, those ones that inhibit DNA and RNA synthesis. And these examples include, we shall say examples like fluoroquinolones, and these fluoroquinolones, they include ciprofloxacin, ciprofloxacin, we have levofloxacin, and these ones, they act by inhibiting an enzyme which we call DNA gyrase. They inhibit DNA gyrase, or what we call topoisomerase 2. Topoisomerase 2. This topoisomerase is that if in case we have the DNA, and we have DNA during replication, Along the replication fork, there is always the formation of supercoils. So these supercoils formed during DNA replication. So there is always the formation of supercoils, and these supercoils are removed by DNA gyrase, which acts as a bulldozer. For it comes and removes the supercoils so that helicase can continue cutting to unwind the DNA double strands. So the role of DNA topoisomerase or gyrase is to remove the DNA supercoils so that helicase can continue to unwind the DNA. So what fluoroquinolones do, they come and bind to topoisomerase, or what we are calling DNA gyrase enzyme, which, so in case the DNA gyrase is not functioning due to the replacement of these fluoroquinolones, then the DNA strands that do not unwind, and when they do not unwind, meaning we cannot replicate to form more copies. That is how the mechanism of cipro and levofloxacin, by inhibiting DNA gyrase that is responsible for removal of supercoils during DNA replication. That is what we can talk about. Then another class, this is class A. Then in class B, we can talk about those that inhibit folic acid synthesis. Many books put this one under nucleic acid synthesis. Why? Because the folic acid is also a precursor of proteins of DNA synthesis. So these ones that inhibit folic acid synthesis, we call them antimetabolites, and they include sulfonamides, and they include trimethoprim. So trimethoprim and sulfonamides are the key or the peculiar examples that inhibit folic acid synthesis. How is that inside the cell membrane, we have PABA, which is the para-amino benzoic acid, which joins with glutamate to form dihydrofolate. And this dihydrofolate is formed in the presence of dihydrofolate synthase, or what you can call synthetase. Then after forming dihydrofolate, it is converted to tetrahydrofolate in the presence of dihydrofolate reductase. So we are going to see sulfonamides inhibiting the dihydrofolate reductase, whereas trimethoprim is inhibiting synthetase enzyme. So, and when we don't form tetrahydrofolate, we cannot form folic acid. And without folic acid, we don't form purines and pyrimidines that participate in DNA synthesis. So that's why we classify it under DNA synthesis inhibitors, because once we don't have folic acid, we do not form purines. Those are nitrogen bases that are responsible for synthesis of DNA. So that is sulfonamides, and some of the examples of sulfonamides include, we have cotrimoxazole, sulfamexazole, all these are examples, and sulfadiazine. Sulfadiazine is also an example of sulfonamides. Sulfadiazine. And sometimes they're always given in combination to prevent the bypass. Under antimicrobial resistance development, we shall see that some bacteria, they develop a bypass mechanism. So combining sulfonamide and trimethoprim prevents that bypass mechanism. Then another part B under inhibitors, part C, we are going to talk about the imidazoles, what we call the metronidazole. We have tinidazole, secnidazole, together with nitrofurantoin, which is one of the leading antimicrobial agent that is susceptible to UTIs, that is nitrofurantoin. This nitrofurantoin, for it acts by these three acts by producing superoxides. oxide, or what we call oxygen reactive species. Oxygen reactive species. These are peroxides that bring about formation of G, which destroy the DNA. So these peroxides, they are the ones that destroy the DNA. Then lastly, which is D, under nucleic acid synthesis inhibitors, we have rifampicin, or what we can call rifampin. And this rifampicin, which is normally given in TB patients, for it is responsible for inhibiting, it inhibits DNA polymerase. And this DNA polymerase enzyme is the one that plays a big role in transcription. That whenever we have the DNA, if this is the DNA, it undergoes replication to form two strands, which undergoes transcription to produce an mRNA. So during the production of mRNA by transcription process, we are saying rifampicin inhibits transcription by inhibiting DNA polymerase that plays a big role in transcription, which is formation of messenger RNA. And without messenger RNA, we cannot form protein. So these ones, we cannot multiply the genome. That is what rifampicin, which is used in the treatment of TB, how it inhibits the Mycobacterium tuberculosis. By inhibiting DNA polymerase, which is responsible or which plays a big role in elongation step during transcription process. After seeing that, lastly, the mechanism we can talk about in this session is protein synthesis inhibitors. And these protein synthesis inhibitors, they target ribosomes. And these ribosomes are divided into 50S ribosomal subunit and 30S ribosomal subunit. The way we are seeing it here, this is the ribosome having the larger subunit, which is 50S, and the smaller subunit, which is 30S. So we're going to see these ribosomes in the cytoplasm. After forming mRNA comes and synthesizes the protein by translation in the presence of a machinery known as ribosome. So what these protein synthesis do, inhibitors, they inhibit the process of translation. They are inhibiting the process of translation, whereby we are converting the codons into a long polypeptide chain of amino acids. So whenever we take some antibiotic agents or antimicrobial agents, those some target 50S ribosomal subunit, which I have said is the large one, and others target 30S, which is a smaller ribosomal subunit. And those that target 30S, we normally have an abbreviation known as AT, whereby A means aminoglycosides and T tetracyclines, and sometimes we can add another one, but it's not so important. We can live with these ones. Then for 50S, we have a mnemonic known as CEL, whereby CEL, S stands for streptogramins. Then the C stands for chloramphenicol. Then E is erythromycin, but whenever I see erythromycin, I think of macrolides. So this one is representing macrolides. So all macrolides act here. Then the first arrow is standing for linezolid. These are oxaronosids. Then we have another arrow which stands for lincosamides. Lincosamides like clindamycin. So clindamycin is an example of lincosamides. So all these, they act by inhibiting 50S. They act by inhibiting 50S ribosomal subunit. 50S ribosomal subunit. And when we go into details, we shall tell you that in mechanism, Apart from inhibiting the 50S, which is the largest subunit of ribosome, chloramphenicol, it inhibits directly the peptidyltransferase enzyme. And this peptidyltransferase enzyme, which is in the larger subunit, is the one responsible for forming of peptide bonds in between amino acid during translation. Whereas macrolides, for it inhibits by stopping translocation of ribosome. As we know that during translation, after adding an amino acid, the ribosome moves one step ahead. Adds another amino acid, it translocates one step ahead. So what macrolides do, that is erythromycin, azithromycin, they inhibit the movement of ribosome. So it becomes stuck in one place and we cannot continue to synthesize the polypeptide chain. So this is what we can talk about under 50S, those ones that target the 50S ribosomal subunit. And those that target the 30S, we are saying we have aminoglycosides. We have given example, those ones that target 30S ribosomal subunit. We have listed, we have said the abbreviation is ART, which means aminoglycosides, and tetracyclines, whereby under tetracyclines, we have tetracycline, we have doxycycline being part of this, and aminoglycosides, we have amikacin, kanamycin being part of the aminoglycosides. So these are the examples, those ones that target the smaller subunit, which is the 30S, inhibiting the protein synthesis. So these are the 4 mechanisms of action of antimicrobial agents, especially antibacterial agents, where we have seen in recap, we have seen those that inhibit cell wall synthesis, that is beta-lactams, the vancomycin and phosphomycin inhibiting the cell wall synthesis. We have seen those that inhibit the functioning of cell membrane, including polymyxin and amphotericin B, which is an antifungal. Then number 3, we have looked at nucleic acid synthesis inhibitors, which we have subdivided them according to their mechanism of action. Their mode of action is inhibiting, but the mechanism is different, whereby we have seen those that inhibit DNA gyrase, that is topoisomerase 2, those that inhibit folic acid synthesis, that is sulfonamides and trimethoprim, and we have seen those that inhibit DNA by production of superoxides, which act as a poison against the DNA. And we have seen finally rifampin, which is responsible for inhibiting DNA polymerase, responsible for transcription. And lastly, we have looked at those that inhibit protein synthesis, that is the process of translation, whereby it occurs in the presence of ribosome containing the larger subunit and the smaller subunit. Larger subunit 50S and smaller subunit 30S. And we have used the mnemonic that those ones which inhibit larger subunit is cell, which stands for streptogramins, chloramphenicol, then macrolides, linezolid, and linconsamides. And then lastly, we have seen those that inhibit the smaller ribosomal subunit, which we have seen tetracyclines and aminoglycoside, which we have said the abbreviation is ART, cell. ART means the smaller, then cell meaning the larger subunit. Thank you so much for following from the beginning until the end. Always remind you to subscribe, like the video, and share to others.